So we'll do a quick review over DNA and DNA structure and talk about the structure of genes. And so just to remind you what a nucleoside or nucleotide looks like, at the core there is a sugar, ribose, or in this case deoxyribose, since we're going to talk about DNA first. And to number the carbons on that sugar, we first need to identify where the anomeric carbon is, and that's that one right there. That, of course, is the carbon that's bonded to two electronegative atoms, the oxygen here, the nitrogen of the base. So that base is attached at that position. That means that this is the 1' carbon, this is the 2', 3', here's 4', and then 5' is here, and that's attached to the phosphate group. So that's the overall structure of a nucleoside monophosphate in this case, or a nucleotide unit of something like DNA. And of course, speaking of DNA, nucleotides can form polynucleotides, that is polymers of these that are connected via phosphodiester linkages, in this case shown right here and here. And remember again, this is the 1', here's 2', 3', 4', and 5'. And so this is the 3' end of the molecule on this end. Because it has this available 3' hydroxyl group, it's not involved in a phosphodiester linkage. And on this end of the molecule, this is the 5' end, because it has a 5' carbon here that is not attached to another ribose via another phosphodiester linkage. And as we indicate, it is conventional to read sequences of polynucleotides in the 5' to 3' direction like that. So if you were to identify what this polynucleotide is, we would say it is 5'. Looking at the nucleotide bases here, this is an adenine base, so that's an A, and then we have a G, and then a C, and then we have the 3' end. So that is the proper way to name this. If we were to name it in the opposite direction, that is 5' CGA, that would be a completely different trinucleotide. And so directionality definitely matters. I'm not going to worry too much about having you memorize the structures of these bases. That's not really the point here. It's more about understanding overall DNA structure, and particularly how it's relevant to the things that we're going to be covering in this course. Remember that bDNA is the most common form of DNA, and that's what we're showing over here on the right. This is the way your genome exists most of the time. Of course, there are other types of DNA's structures that occur during replication and those sorts of things. Remember that they're stabilized by pi stacking interactions, and so that's what's sort of holding all of this together. There are, of course, hydrogen bonding interactions that are sort of provide the specificity for AT and GC base pairing. So, of course, these groups here, here, and here are responsible for the base pairing interactions that we see. Now that we're refreshed about what DNA structure is like, let's talk about genes and then also transcripts. And the first thing I want to mention are sense and antisense DNA strands. And so by definition, the sense strand is the DNA strand that contains a gene sequence such that the beginning of that gene is at the 5' end, and then the cessation or the end termination of that gene is at the 3' end. This is also called the coding strand, and we call it the coding strand because the DNA sequence is sort of occurring in the same direction that the final protein sequence, amino acid sequence, will. So for example, this would be if we're saying that the gene starts with the amino acid A and goes B, C, D, so we're just doing these in alphabetical order to indicate that A is the beginning of the gene, that the A is at the 5' end, right, of the sense strand. And that the 5' end of the antisense strand would be the end of the gene right here. Whereas the 3' end, right, is the end of the sense strand for that gene. And so another example, we could have on the opposite strand of DNA, potentially right next door, another gene that has its sense strand going in the on the opposite strand. So here we have the 5' end beginning of the gene, A, B, C, D, etc., and then the end is over on this end. And so the antisense is what the opposite strand is. The antisense strand, we also call it the non-coding strand, but it is actually the strand that does serve as the template when messenger RNA is produced, because that messenger RNA needs to have the same sequence with T's replaced with U's, and of course, as the gene does, as the sense strand of DNA does. And one thing I'll point out is that these are gene-specific, not globally. So what that means is that a particular stretch of DNA, like this could be a part of a chromosome here, can contain both sense and antisense on a particular strand, as shown here. So it's specific to the gene. Let's talk about what we would see in a typical gene transcript. Another term for that would be messenger RNA. And it's more than just a gene sequence, because there's sequence that doesn't end up in the protein. We call those the untranslated regions, and if it is on the 5' end of the gene, we call it the 5' untranslated region, or UTR. And if it's on the 3' end, we call it the 3' UTR. And there's some useful things in the 3' UTR, but we don't really talk too much about in this course. The 5' UTR definitely contains more useful things for what we're thinking about. The transcription start site is at the +1, and there's dozens of base pairs that we go along until we get to this Shine-Dalgarno sequence, also known as the ribosome binding site. And it has roughly this AG-rich sequence, and then that is really just a few base pairs before we get to the actual start codon of the gene, and that sort of lines up things properly at the ribosome. And then we have the gene after that. So let's talk about the translation of that messenger RNA. And we should know that each amino acid in a protein is encoded by 3 nucleotide bases, and those are of course known as codon, and a gene's going to have a start codon. It's usually ATG, which will code for methionine. Once in a great while, especially in bacteria, we might see that GTG is the start codon. That's valine, but vast majority of the time it's an ATG start codon. And there's going to be a bunch of codons in between for amino acids, and then we will see a stop codon. That's the end of the protein. So here we have a start codon, a bunch of bases that code all of the amino acids of the protein, and then a stop codon that signifies the termination of translation. And of course, there are various codon tables we can look up to identify what a particular 3-base codon amino acid is equivalent to. And so a lot of times we do see U's instead of T's in this table, because of course it is the messenger RNA that goes to the ribosome to enable translation. So a lot of times these tables are looking at U's instead of the T's that we would see in the DNA sequence. And then this leads me to finally talking about reading frame. This is the way that DNA or RNA is sort of broken up into those 3-letter codons. And there should make sense that there's going to be 3 forward reading frames and then 3 reverse reading frames. And so for example, let's look at the sequence shown here. We can break that up into multiple different ways, where we just take the first 3 and call that a reading frame and kind of go through that register. We could instead take the first 2 and then start a codon after that. Or we could just take 1 as a gap and then take the following codon after that one. And this is important because this has big consequences on the protein sequence, and you can see if I use the clustering of 1 down here, I get a very interesting sequence that has a stop code on it. That could be a problem if this is the middle of a gene. And furthermore, as we change the reading frame, you can see we very clearly change the sequence of the proteins. And here I've only shown the forward reading frames. There, of course, are reverse reading frames where we would read this that way, but we typically think of these only in the forward direction because we're normally identifying sequences from the 5' to 3' direction. But if we were looking at instead a non-coding strand, the antisense strand, we might consider the reverse reading frames as well. But for our purposes, we're pretty much always going to be looking at the sense strand. And so an open reading frame is a piece of DNA, and it's going to have a start codon that you can identify. There's going to be a long stretch of codons where we do not encounter any stop codons, something on the order of a couple hundred to a couple thousand base pairs, and then a stop codon. And so if you see that, start, a bunch of codons, and then a stop codon, then we know we're probably looking at a protein, and those are typically referred to as open reading frames.